why don't we have you all make sure to stay muted so there's nothing going on in the backgrounds. You guys have that question sheet to work off of, so maybe have that open in the background. But of course, you are welcome to ask questions just that pop into your head as well. To ask questions, why don't you raise a virtual hand, and then I will call on you and you can ask. We'll also have some questions potentially coming in from the live stream on Facebook. So Dr. Troutman will send me those questions, and then I will ask them. So any questions? Everyone feel good about knowing how to raise a virtual hand? Okay, I see thumbs up. Good deal. Wonderful.
Just one other note, if you want to send in a question through chat, that comes to me, and then I'll pass that along to Mrs. Pieroni. So, hi to all the students that I haven't seen in a while. I hope everything's going well. All right, whenever you're ready, we are good to go.
Okay, hello everybody. I am Heidi Pieroni, teach AP Biology at Thomas Jefferson, and we're here with AP Biology and our two experts to open up a discussion about COVID-19. So let me welcome our experts and introduce them. First, we have Richard Levy, who has more than 38 years of experience specializing in endocrinology, diabetes and metabolism, and pediatric endocrinology. He is double board certified in adult and pediatric endocrinology. Laurie Shornick is associate professor and biology department chair at St. Louis University. Some of the courses she teaches are advanced principles of virology and concepts of immunobiology. In the Shornick Laboratory, she leads research exploring the neonatal immune response to respiratory viral infection. So before the Q&A portion, we're going to start with getting some information from our experts, and then we'll move on to the Q&A portion. So Dr. Shornick, I'll turn it over to you.
Sure. So thank you for having me. And I have just a couple of quick slides here just to sort of lay the groundwork, and then we can answer some questions. And a lot of times people just want to know what a virus is. And so this picture is showing you the SARS-CoV-2 virus. And you can see it's sort of this green circle with some lighter green little spikes sticking out of it. And that's actually where it gets the name, because when they first looked at it under the electron microscope, they thought it looked like a corona or a crown on the virus. And so those things that are sticking out are proteins. They're called spike proteins. The green part in the middle is the genome of the virus. So this virus has a genome of an RNA molecule, whereas in our cells we have chromosomes with DNA. This virus has RNA that's protected by a nucleoprotein, which is actually shown in green. And then it has a lipid envelope that it takes from the host cell. And what I want you to kind of focus on is this big tan box on the bottom is showing you a cell. And the virus is coming up to that cell and it's going to attach. And that's always the very first thing that a virus wants to do. It wants to attach to our cells. And in this case, the spike protein on this virus binds to a receptor or a protein on our cells called ACE2. And that's going to help it attach. There's another protein that's coming in that will cleave the spike protein, so the virus membrane can fuse with the membrane of the host cell. And then you can see the virus gets pulled into the cell. If you look down here, we have something called the late endosome, and then we have the viral genome opening up, being released into the cytoplasm of the cell. And then the virus basically uses the cell's own machinery to make copies of itself through a very highly regulated process. And then eventually we get more viruses. If you look over on the right side where it says budding bicellular exocytosis, we get newly made viruses that bud off of the cell and are released. So that's sort of the life cycle of the virus. We get these coronaviruses by breathing them in, and they're attaching to the cells in our upper airways. As they replicate and make more viruses, the infection moves down from the upper airways to the lower airways. Let's see. So I just wanted to hit on a couple of ideas that you probably hear about in the news, and people are out talking about, you know, getting immunity to the virus. And I just wanted to sort of touch on what it means when we say we're immune. It means that you've already seen this pathogen or this virus before, and you've had a response to it, and you've generated memory lymphocytes, so memory B and T lymphocytes. If we have these memory cells, then they can jump into action right away. Memory B cells can divide quickly and produce a lot of antibodies to protect us from the infection. And what the antibodies do mostly is they're actually going to coat the surface of the virus so it can't do that initial attachment step. So that's the main way that antibodies are protecting us. And then I wanted to also talk briefly about what immunization is. So there's a couple ways that we can become immune to something. There's active immunization and passive immunization. So active immunization is when you actually develop a memory response, and this can happen either when you have an infection and you recover from it and you develop memory cells, but it also happens when you get a vaccination. And when you get a vaccination, you mount an immune response to the vaccine and you develop memory cells to protect you in the future from that same pathogen. Another type of immunization is passive immunization, where you take antibodies from one individual and transfer them to another individual. The natural version of this is with mothers and infants who transfer antibodies from their blood to the baby during gestation and then through mother's milk during breastfeeding. But we also sometimes do passive immunization for people who are immunocompromised. So they might get treatments of antibodies from other people to help protect them. And that is one of the things that we're looking at as a treatment for patients with COVID-19. When we're talking about vaccination, there's a concept called herd immunity that's important to understand. So in this picture, each dot represents an individual within a population. Those who are red are not protected. The folks that are blue have received a vaccine and are protected. And the folks that are the yellow dots are susceptible. So if you look at the top left, you can see where we have 10% vaccinated. Many people in the population are not protected. And so that means the pathogen could be transmitted among that population, and those susceptible people in yellow have a high probability of encountering the pathogen. As you increase the vaccination rate, as you go across the top, you can get up to 68%, and then we continue along the bottom, we get up to 99.7% vaccination rate. Where most of the people are vaccinated, then you don't have the likelihood of infection being spread throughout the community. And those yellow individuals who are most susceptible, and this could be people who are immunocompromised due to chemotherapy, due to a genetic immunodeficiency, and other things, that helps them be protected. So we want to have good herd immunity. We want to have most people protected in order to prevent spread of infection. A couple of slides I added in just really to help answer questions. So I can end there, and then Dr. Levy can talk, and then we can take the questions.
Okay, I will get that set up.
I'll stop screen sharing here.
Yep. All right.
So I'm going to try to answer several questions, but there's some repetition in the basics, and I'll kind of fly over those things. So what is COVID-19? Where did it come from, and what does it do to you? are the questions that, that Heidi asked me to address. Why don't you go ahead? So this is a electron micrograph, and you can see the virus particles there. And this is the usual type of picture that that an electron micrographer would have. Why don't you go to the next slide? Can be color enhanced, and if you take a look at this, you can see those little spikes coming out if the arrow works there. And that's where the name comes from, coronavirus, because corona means crown-like spikes. Let's go to the next slide. And Laurie addressed that this is the genetics. And if you recall her picture, there are many proteins that are manufactured and then they have to be reassembled. It's a very complex virus and it's one of the largest. It's about 32,000 bases or nucleotides, and it's a huge RNA virus. There are DNA viruses and some of them can be larger than the RNA viruses. And this is a cutaway of the viral molecule, I guess you'd call it. And you can see various components. Here's the spike, for instance. You have protein in the envelope. You also have protein in the capsid here. You have lipid here as well. So essentially, it's not that complex a structure. And actually it's kind of pretty. Why don't you go ahead to the next slide? And getting back to the genetics, you can see that the components have areas that program for the various components of the virus, envelope, nucleocapsid, and so on. Let's go to the next slide. This is an atomic level structure showing the spike. This green up here is the spike, or the crown if you want to. That's how it attaches and gets in. We'll go into that in a little more detail. Next slide. And the virus, the spike, depends on essentially anchoring or attaching to the angiotensin converting enzyme. This ACE2 receptor is also involved in treatment for hypertension. It was not known whether the ACE blockers would have a good effect or a bad effect. If anything, it seems to have a good effect, but it's negligible. But it's interesting. You also have, better go back. Okay, you also have essentially a protease that activates the spike protein. So you need both of these, and go right ahead. This is a picture of the protease area right here. And essentially that's another area that could be attacked by a monoclonal antibody or a vaccine or something. So there's more than one way to get at this virus. Go ahead. Not to belabor this, this is the spike protein that we saw, but essentially you have domains as three of the viruses, so to speak, get together and attach. Here's the spike, and you have receptor binding domains that are down, and you have one that is up, and that's the one that will attach. It's not unusual for there to be dimers or trimers when various molecules attach to a membrane. It helps them get into the stereo position. And you can see here's the ACE right here, and the protease domain as well as pictured. So it's another way of looking at the virus in terms of its configuration in space. Why don't you go ahead? I think Laurie pretty well went into this in terms of the attachment and the replication. Obviously, what the virus is doing is taking over the cell machinery and making more virus. And so essentially, that's how you get sick, and the virus propagates itself. And we go on to the next. This is a phylogenetic tree in terms of answering that second question as to where the virus came from. And essentially, it looks like it came from bats. So this is a zoonosis. It existed in another species. You've heard a lot about avian flu, and so that came from birds. Why don't you go to the next slide? There can be an intermediate host to the bats. In other words, the pangolin was supposed to be the intermediate host that was brought into the Wuhan fish or wet market, and they would sell wild animals for food and so on, and that's how it is thought to get to man. Obviously, we've been transmitting it to ourselves, and Laurie showed you a map of that. This is how it appears to have been acquired, but right now we're simply giving it to ourselves. We don't need the animals anymore. Let's go ahead. We were hoping that the virus would not mutate, and it seems to do less than the flu, which is highly variable. The influenza is so mutagenic and has so many genetic shifts that every year a trivalent or even a quadrivalent vaccine is needed to try to predict which strain to try to prevent people from being infected with. But as the virus has spread, there's at least 3 major strains and maybe 5 or so more, and at least a couple of dozen different mutations that have been found. This is, again, much less than the flu. Why don't you go ahead? This I thought was kind of interesting. This is a protein that is made by the virus to make it appear more like normal tissue. So it kind of masks itself. And it's one more area that, since it's been identified, that could be attacked in order to fight the virus. Let's go ahead. I like this picture because it shows the virus in relationship to human tissues. And it's essentially with a scanning electron microscope. And it sort of introduces the last question that I was assigned, and that is the clinical presentation. Laurie mentioned that it comes in via the respiratory system. It can aerosolize, and the question of transmission is something high on everybody's mind. But it's where these small particles, and a virus, at least this one, is about 0.125 microns. If you have a very small smoke particle, for instance, it's around 2.5 microns, and larger ones can get larger. And even the 2.5 micron smoke particle gets down to the alveoli. So you can see that something that is about 20 times smaller easily can get down to the air sacs, the alveoli, and infect and cause a pneumonia. So you have variation in terms of what the presentation is going to be. It looks like the greatest number have fever and cough, and essentially upper respiratory type of symptoms. Some have no symptoms at all, but you can have some GI symptoms, and you can have myalgia or arthralgia, that is muscle and joint aches and so on. And atypical presentation, if you will, is simply not uncommon in the flu as well. In fact, flu-like symptoms are pretty much very close to what you can get with this virus. Why don't you go ahead? If you do have pneumonia, you have some typical appearance. This is a 37-year-old man, and the arrows here are pointing to what would be called a ground glass type of presentation. It's not that that's unique to this virus. I have another slide I didn't include that shows vaping, and with vaping you can get a ground glass appearance as well. So it's just something that this particular virus can cause. Once you go to the next slide, in fact, it is much more visible here in terms of what you're seeing with this ground glass appearance in a 63-year-old with a severe type of appearance. Reticulation just means a network type of appearance, and between the ground glass and this reticulation, you can have involvement of whole lobes of the lung. And this is a picture of the various phases: early infection, a pulmonary phase with shortness of breath and hypoxia, where you have low amounts of oxygen getting. And obviously, once what's called the chemokines are released by those cells, such as B cells, these chemokines can cause an inflammatory response that overwhelms the body and can cause a lot of fluid exudation and acute respiratory disease syndrome or distress syndrome, shock, cardiac failure, multi-organ failure, including the lungs, hematologic type of abnormalities with clotting and disseminated type of coagulopathies. So all of these types of complications have to be addressed in very severe cases. Why don't you go ahead? This is the critical care management of a COVID patient from here in Chicago. It can get pretty complicated, but the whole idea is you do everything in your power to keep the patient off of a ventilator, oxygen, putting them in a prone position to mobilize different areas of the lungs, reducing that inflammation with the chemokines. Because once they do get on a ventilator, about 4 out of 5 don't get off. And so it's really a last-ditch type of treatment. Let's go ahead. There's a higher mortality rate with some segments of the population, those that have chronic cardiovascular disease, you know, like heart failure and so on. Those that have diabetes, which I deal with, high blood pressure, chronic respiratory disease, cancer, much higher mortality rate than you would expect in the general population. And with kids, it's even less, although children have been now shown to sometimes come up with an equivalent of what's known as Kawasaki disease, where you have a lot of tissue swelling, rash, high fever, that sort of thing that you would not expect to be in such a high percentage, because it's a fairly rare type of disease, but it's a manifestation in some of these children. So they're not exempt from getting the virus and getting a severe outcome. Just go ahead. And that is the end. I'm sorry it was a little bit longer, but if there are any questions, and we have a list, I'll be glad to try to answer them. Was that a dog of a presentation or not?
That was lovely. Thank you very much. Okay, well, students, we're gonna open it up to some Q&A now. If I could just maybe request that for the question and answer portion, you turn your cameras on so we can see your lovely faces as you ask questions. We would appreciate that. Thank you. All right, well, raise your virtual hands and I will call on you and we can get started. Thank you very much for your presentations.
Mrs. Pieroni, we did have a question come through. If you take a look, I sent it over in the chat. Yeah.
Yeah. All right. Well, this is for Dr. Shornick. Does herd immunity only come from vaccinations, or can herd immunity be achieved through some other way as well?
So you can achieve a memory response and immunization through either a vaccination or active infection. So actually that's something I think that Sweden has tried, is they kind of stayed open, thinking that they would have develop herd immunity. Of course, that you risk losing more people that way. Doing a vaccination is obviously safer if you can make immunization without making people, giving them that memory response without making them sick or having deaths. So.
Thank you. Here we have another Facebook question. What would happen if you transplant healthy lungs into a COVID-19 patient? Dr. Levy, would you like to take this one?
I'll try. The question would be when. It would be counterintuitive to transplant a healthy lung into someone who has active disease, because for one, they wouldn't tolerate it, and number two, it's an infected host. But if you're talking about a lung transplant to someone who has gone through the disease and is now essentially wasted, and some of the ventilator survivors are in that position and probably will be for months, I could foresee way down the road that being a possibility. But you know, we're nowhere near that stage at this point.
Okay, Drew.
Marissa had her hand up before me, so I think it'd be fair if she went first.
Okay, we'll turn it over to Marissa. Drew, you can follow up after her.
Thank you, Drew. I was just curious, Dr. Shornick, I know that in your introduction, we talked about a little bit about how you've researched with infants and their response to viral diseases. And I was just curious as to how in recent, like very recent developments of COVID, as we've seen, have dealt more with children and younger people. So I was just kind of curious as to how your kind of research is, if it's changing to kind of, I don't know how to explain this, but what are the effects that you're noticing in not only your research, but how you're trying to gear that towards some sort of a conclusion, if that makes sense?
Sure. So infants, when they're born, they don't have a fully developed immune response. They don't, in most cases, don't have as much inflammation. And they have a different type of immune response. So one of the slides I put in to help answer questions was to let you know that there are different types of immune responses, sort of three main types, depending on what pathogen that you have. And infants and young children tend to have more of a type 2 response, whereas adults that get a virus tend to have more of a type 1 response. In my lab, we study respiratory viral infection in mice because we don't want to be studying human infants. And we use an RNA virus that has a lipid envelope. It's not a coronavirus, but it has some similarities. And when we give that to adult and neonatal mice, we see that they're both able to clear the virus or get rid of the virus from the lungs with the same efficiency and with the same time course. What we found very interesting was that the infant mice could do that without any inflammation in the lungs. And so with these respiratory viral infections, the problem with the lungs is that it's actually your immune response that's causing the problem. You have a lot of white blood cells that come into the lungs, you have a lot of mucus production, and that fills up those very delicate air spaces so you don't get good gas exchange anymore. So we found that the infant mice in our model could get rid of the virus without all of that white blood cells and mucus coming in. And so we're trying to figure out what the mechanism of that is, because that would be a great therapeutic if you could block the inflammation without hampering the ability of the body to get rid of virus.
Cool, thank you.
You're welcome.
Thank you. Okay, Drew?
All right, so Dr. Shornick kind of answered my question, or at least gave me a bit more insight, but my apologies if this was already answered earlier in the presentation. I got here a little bit late, but what I'm most curious about is how, like, the difficulties that arise with developing a vaccine, because while my understanding isn't the fullest that it could be, what I understand is that we develop antibodies based off of like a dead version of the cell. And so I'm curious how creating a vaccine is taking a long time, and like the difficulties that arise with handling a new virus.
So there's a variety of different ways that we can make a vaccine. The best vaccines are ones that cause a little bit of inflammation. So we have some vaccines that have been very successful historically that are what we call attenuated vaccines. So these are actually viruses that are weakened, so they're hopefully not going to cause disease, but they can still replicate, and so they're going to initiate a really good immune response. And then we can go to the other end of the spectrum where we make just a single recombinant protein, like we make a recombinant spike protein and we use that to immunize. That wouldn't cause as much inflammation. It might not mount as good of an immune response. The making of the vaccine is pretty quick and easy to do, but then figuring out which ones are really efficacious, which ones really work well, figuring out the correct dosing, whether you need booster shots, all of those things take a lot of time, and they have, you know, ideally we would be testing them in animal models first before we test them in humans. So, you know, the initial design, there's a lot of different vaccines for this virus already in the pipeline, but figuring out which ones are going to work best takes time.
All right, thank you.
Sonora?
All right, so I also have a question for Dr. Shornick. So based off of the concept of herd immunity, do you see COVID disappearing as a virus, or like becoming more of a common virus like the flu or something seasonal?
I think we don't know yet, because we don't know whether people who have been infected the virus have long-lasting immunity to it. If it's something like, say, for the most part, and this is generalization, but for the most part, if somebody gets chicken pox, they don't get chicken pox again later on. But we don't know if having this infection gives you that long of a immunity. I mean, it's only been around for, you know, about 6 months or so, or 7 months. So that's another issue. And then the same thing applies to the vaccine. So we need to have some information about what will confer long-term immunity. Otherwise, I do think it will be, it'll keep coming back. Yeah.
Thank you.
Mm-hmm.
Hey, Kira?
Okay, so can y'all hear me? I don't know if it was on mute.
Yeah.
Okay, sorry. I wanted to know what are the best measures to take in order to prevent misinformation, because as we all know, a lot of our media outlets aren't typically becoming TV news media anymore for the younger generations. And I've seen a lot of misinformation going on different platforms, being Instagram, Facebook, and other such. And a lot of people reposting and resharing it, which of course creates a huge problem. And sorry, this is kind of a two-part question, but also the second part is, should the government allow the reopening of businesses and beaches and other things for people to congregate in.
Dr. Levy, you want to
Sure.
take a look at this one?
Well, as you're pointing out, this is politically sensitive. So initially, the whole idea was to, quote, flatten the curve, and that is to prolong the interval that people came down with the virus so that they wouldn't be such a rush and a pressure on the healthcare establishment in terms of equipment and personnel and so on. Ventilators were prominent in that calculation. And to a degree, that has been successful because except for certain areas, you know, like what happened in New York, the number of ventilators and the beds that were occupied have been kept in manageable proportions. Then there's the question of whether or not that having been largely accomplished, that the curve has been flattened, whether you can open up for reasons of the economy in terms of allowing people to get together without their social distancing or wearing a mask and things like that. And certainly, we've been warned that the more we do that, the more resurgence of the infection rate you're going to see. And there's just the question of whether that's acceptable. I think what you're going to see is that the population will roughly divide among those that feel that they have the freedom to do what they want, no matter what the consequences, and those that are more fearful of what those consequences are going to be. And they'll hang back while the others rush to reestablish their normal life, economic included. One way or the other, whether we have a vaccine sooner than later, and it's been predicted more for 12 to 15 months than it is for something by the end of the year or whatever those that represent that it's going to be sooner. Whether you have a vaccine or not, there's going to be those that will climb in terms of having the disease and having some degree of immunity. I'm rather just sanguine that there will be more of an immunity and that hopeful there are not too many strains of the or mutation or genetic shift or any of this type of thing that causes different mutational strains to emerge, and that when you have this, that you don't have the flu-like situation of having to have multiple strains represented by your vaccine, and that you don't have the ability, having had one strain of infection, to have another strain the following year. We can see that with the flu, so I'm just hopeful that's not going to be the case and that you'll have more or less a long-lasting immunity and not have that type of exposure to different strains going onward.
Do we know what the R naught for this virus is? Like how many people are going to be infected from one person who has the virus?
We do, Kyra. It's about 3 to 5. So it's much more than influenza and much less than measles. So to go back to the original part of your question, you asked what sources to go to. I recommend the CDC website. I recommend the World Health Organization website and scientific journals. So if you go to Science or Nature, they all have pretty prominently now a COVID section, and you can go to that and find up-to-the-date information about the virus. So I just put up, this is from the World Health Organization, the number of cases worldwide. This is from today. And you can see that there's a very large circle indicating the number of new cases over the US. And one of the reasons it's so large here is because we haven't been doing as well with sheltering in place and social distancing as other places. So those things are going to be important for keeping the number of infections low as we move forward. And it's pretty easy to follow the news that wherever someplace opens up, about 14 days later, there's an outbreak in that location. So there's a graphic from the CDC that shows that there was some kind of social function of over 50 people. One person there was infected, and then a whole bunch of people got it, and several died from that. So it's important to, you know, when you are out, wear masks. We know that it's transmitted via droplets, but also by aerosols. So droplets are bigger particles that you would get when you cough or sneeze. So they would be actual moisture droplets that could have millions of viral particles within them. But in fact, just aerosol, when you talk or breathe, can also pass the virus. So that's why it's important to wear the mask.
That episode of Super Spreader happened to be a choir that got together. And
right, yeah.
You can't get better than that
exactly.
situation.
Yeah, singing is not a good idea.
So to follow up on that, what are the best ways, or what do you recommend when we are out in public to protect ourselves and those around us?
Well, you know, I mentioned that the virus is very small, and Laurie was mentioning about the aerosolization and the fact that if you have a mask, it's better than nothing, but I don't think you should delude yourself that that it's going to be fully protective. At least it would stop you or someone else who's wearing it from having large amounts of droplets being propagated into the air. But it's better than nothing. It's not an N95 mask, or they have even finer pores to try to stop the virus. And mostly those types of masks are being used by healthcare personnel, and they're in short supply. In the hospital that I work at, regular surgical masks are used just as well as those that are caring for COVID patients, that the more effective PPE or protective equipment is being reserved for. That's the best I can say for wearing the mask, that, you know, it might be something that's caught in the fibers of the mask. How about you, Laurie? What's your opinion?
Yeah, I think the mask is very important if you're out and in anywhere, going out of your house, for sure. And cloth masks, you know, if you don't have the surgical mask and you have a homemade mask, that's good too, as long as you have something covering. Yeah.
Again, it's better than nothing.
Yep.
Thank you. We have a Facebook question from Mrs. Roth. What's your take on the future of using convalescent plasma to treat COVID-19, or to use it to protect frontline workers?
Well, I'm very high on convalescent plasma because it seems to work. And a little story, I had some sort of flu-like episode in February, and I wanted to get a serologic test to see if I had the disease. Testing is two different kinds of tests. There's a reverse transcriptase preliminary chain reaction type of test. It's for active infection only. But as it turns out, the serologic testing is unreliable. The new Abbott test is shown to have some major reliability flaws. Perhaps the WHO and Roche tests are the best in terms of quality for the serologic test. And that test, regardless, is being reserved for those who are documented to have had an active infection and are willing to give their plasma to treat very severe cases with it, to show that they have a good antibody titer. So that's a long way of saying that if someone is documented by having had an active test and they're willing to give their plasma, that it's been shown to be quite effective as rescue for severe cases.
So just one little note about that. It's most effective if it can be given early in infection, because you're transferring the antibodies passively, as we talked about, and the antibodies are going to work by binding to that spike protein and preventing attachment. And so you want to give those antibodies at the early point of infection when the virus is replicating. If you give it later on, after the virus is already replicated and you've turned on all the inflammation and the immune cells coming in, it may be less effective. Yes,
but I think you know that, how would I put it, as logical as what you just said, somebody early on would not be eligible to receive it, at least at this time.
Right.
In other words, it's not going to go the way that science dictates when it comes to being able to get convalescent serum. Unless you're Bill Gates or somebody like that.
Okay, thank you. AP Bio students, what do you guys think? Okay, I'm going to steal one of the questions that they wrote for you guys. I apologize from whoever I'm taking this from. We had a question about sort of the weather. So the flu seems to naturally go away in the summer and warmer temperatures. Viruses, some of them seem to prefer colder temperatures. So is this something we might see naturally die down a little bit as temperatures go up?
Well, I just put the map up. So you can see that there are viral outbreaks throughout the world in some places where it's already very hot. So I think that answers the question that it's probably not going to be as seasonal as some other viruses that we see. And we can't say for sure because we haven't been through a full year or full season yet, but just the fact that it's in cold, temperate environments and, you know, all different kinds of environments right now. So that's my take.
I think what you're hearing is a lot of wishful thinking that's perhaps politically based. And, you know, miracles happen, but to depend on them is not a great idea.
What is it about some viruses that make them not as prevalent in warm weathers? Is there anything we know about this virus that makes it different, or is it just what we're seeing?
Well, it is true about the flu. It's a big spike in February for the influenza virus. But you still see flu cases throughout the year. So I really don't know the answer as to why it sort of dies down and then picks up at that period of time. Laurie, do you have any insight?
Yeah, I mean, I think when we talk about this with viruses, there's a couple of factors involved. One is people look at things like humidity, and I mean, some viruses may be affected by higher humidity or lower humidity, and that may affect droplet size. But also it's human behavior. So sometimes we're in more close quarters, or we're all back together in school, so there's greater transmission, as opposed to summertime when people are not, you know, they're out more and not in close quarters together. So it can be a mixture of things, and every virus is going to be different.
Just to be fair, this is not the only seasonal variation in a disease that you can see. Diabetes that I deal with, type 1 diabetes, also has seasonal variation. So it's of some interest, but you know, once more, you can get it at any time.
Yeah. Marissa?
Okay, so I was just curious. So we haven't seen this strain of coronavirus before within like our populations. So I was just, I was trying to understand, like, how much do we know about the coronavirus that's currently spreading, and how do we expect our knowledge to kind of change with time going on?
Knowledge of what?
Like, I don't know how to articulate it, but do we know like what the long-term consequences of the virus will be based on the people who are recovering, or are we?
Oh, for an individual that has the virus?
Yeah, like if you survive from it, do we know what, if at all there would be long-term consequences that result from it?
I think there will be long-term consequences, because a lot of these respiratory viruses actually change the lungs, and so people that recover may have long-term respiratory issues, whether it's, you know, asthma or COPD or something like that. The virus that I work with in mice, so one infection changes the airway cells of the lungs so that they make more mucus all the time. So I think that's definitely a possibility. And we'll just have to follow these patients out and see what happens.
Okay, thank you.
Mm-hmm.
Okay, any final questions out there? Okay. Well, thank you so much, Dr. Shornick and Dr. Levy. We really appreciated your time and your presentations and your expertise. You know, personally, that really clarified a lot of the questions I had. So thank you for joining us today.
Oh, you're welcome. Thanks
Welcome.
for having us.
Sure.
So wear your masks and wash your hands, everybody.
Definitely.
And keep 6 feet away from me.
All right, thank you.
Okay, thank you so much.
Take care.